Hello everyone. A very good afternoon to you all. I am Himani. I'm the brand marketing manager here at Cambrian Bioworks, and I'll be your host for today's webinar. We've got a really fascinating session lined up today. Our focus today is on male infertility diagnostics, a space where traditional methods have often fallen short, but where emerging genomics diagnosis are beginning to make a real difference. We're joined by Dr. Harsh, a clinician and a researcher whose work is helping transform the way we approach male infertility through targeted genomics. This session is part of our ongoing monthly webinar series. If this is your first time joining us, welcome. We started this series to create a space where labs and clinicians can learn from real-world case studies, share practical insights, and connect with others working in the frontiers of genomics. So far, we've covered topics including stool DNA extraction for microbiome research with leucine-rich bio, selecting the right beads for NGS library prep, and whole exome sequencing in clinical practice with Strand Life Sciences. If these are the kind of sessions you'd like to follow, feel free to subscribe to our series. You'll see a QR code on the screen. It'll take you to our events page where you can subscribe and stay in the loop for our upcoming sessions. Let me introduce you with our speakers for the day. It's an absolute privilege to welcome Dr. Harsh Sheth. A very warm welcome to you, Dr. Harsh. He's the head of Advanced Genomic Technologies Division at FRIGE's Institute of Human Genetics in Ahmedabad. He's also the founder and CSO of Decipher DNA. His research spans across rare disease diagnostics and precision genomics. He's been leading the development of NGS panels for male infertility diagnostics. He'll take us through this in detail in today's session. We're also joined by Diksha Suresh. She is the product marketing manager here at Cambrian Bioworks. She works closely with both our users and the development team to bridge the gap between the lab needs and the product design. She'll take us through how this workflow can be implemented seamlessly in a real-world lab setting. Dr. Harsh will start by walking us through the current diagnostic landscape in male infertility, including where traditional methods often fall short and clinical challenges labs commonly face, how next generation sequencing is reshaping this space, with a closer look at targeted gene panels and their diagnostic value, the clinical implications of a more precise diagnosis, and how better insights can guide treatment decision and improve patient outcomes, what new diagnostic paradigm looks like, and how it compares with conventional workflows in terms of accuracy, speed, and usability. Then Diksha will take us through how your lab can optimize its workflow to implement this approach effectively. We also have live Q&A session, so feel free to drop in your questions in the chat box as we go along. With that, I will now hand it over to Dr. Harsh.
Thank you, Himani, for the wonderful introduction. Good afternoon, everyone. It's a pleasure to be here with all of you. And today I just want to share with you the paradigm shift that is happening in male infertility diagnostics. So, guys, I actually wear two separate hats. One is that of an assistant professor and head of Advanced Genomic Tech Division at Institute of Human Genetics based in Ahmedabad. The second one is that of the founder and chief scientific officer for Decipher DNA Private Limited, which is also based in Ahmedabad. And what we are working towards is to try and understand molecular epidemiology of different diseases. So it may be rare diseases, it could be common genetic disorders as well. But based on that, we start developing diagnostics and very, very soon therapeutic interventions as well. Now, like any good presenter, I begin with a conflict of interest statement. So a lot of work that I'm presenting today is actually covered by a national patent. So for example, I'm named as an inventor on two national patents describing technologies that helps in detecting SNVs and CNVs and gonosomal aneuploidies, all of them simultaneously. I'm also a panel member of ICMR's Task Force on Precision Oncology in South Africa's Open Genome Project. Now, I've spent nearly 11 years of my life in this beautiful place known as Newcastle upon Tyne, which is in the northern part of the UK. So if you guys are reading newspapers quite fervently, then very recently you would have come across 3-parent baby IVF technique. So that was discovered in this very building. It's the Center for Life. It was discovered on the third floor, and I was working on the second floor. And after 11 years stint, I came back to India and I settled down in Ahmedabad, which is also my birthplace. And this is the institute where I work. And throughout this nearly 16 years journey that I've been in clinical genetics, I've had the pleasure of working on various different things like pharmacogenetics of orphan or rapid molecular diagnostics with a company known as Quantum DX, working on understanding the genetic architecture of male infertility, working on cancer prevention, identifying hereditary cancer syndromes, lysosomal storage disorder, developing India's first biobank for lysosomal storage disorders, and developing a whole host of targeted NGS-based assays just for India. But today, what I really want to focus upon is male infertility, because this is one problem that is actually very, very close to our hearts, but we don't talk about it. There's a lot of social stigma about it. And I begin with this very, very simple viewpoint, which was written by E.F. Schumacher in his 1973 book known as Small is Beautiful, The Economics as If People Mattered. And what he states is actually fairly true. Any intelligent fool can make things bigger, more complex. It takes a touch of genius and a lot of courage to move in the opposite direction. And I hope that by the end of my talk, I'm able to convince that what this gentleman was saying way back in the 1970s is actually quite true. So let's begin with infertility. It's in fact a very, very common problem. It affects about 1 in every 6 people worldwide. Half of it is attributed to male infertility, or about 1 in every 30 male in the world is infertile. If you put that in broad context, in India, that would equate to approximately 1.2 crore males being infertile as of now. And genetic factors are estimated to account for approximately 1 in 4 infertile males. That means there is a very high genetic burden that's running in the background. And when we think about infertility, they come in 2 broad flavors. The first one is quantitative defects. So that's simply looking at how much sperm is there in the semen. So you can have anywhere from normal zoospermia, that means you have more than 15 million sperms per mil of semen, or azoospermia, which is the most extreme end, whereby you have absolutely no sperms in the semen. The second flavor is more like qualitative defects. So this is where the amount of sperms are produced in adequate numbers, but the quality or the morphology of the sperms are all over the place. So they might have head defects, they might have tail defects, they might have mid-piece defects, or they might not just have head or tails at all. So in these 2 broad categories is the male infertility subtype divided. Now, since 1990s, the recommendation of the World Health Organization is, if you want to identify that there is a genetic defect in an infertile male, the first thing you do is karyotype to look at the most common disorder, that is Klinefelter syndrome. So this is where you end up with 47XXY. You have an extra copy of the X chromosome. But if your karyotype is normal, then there is a second test you must do, which is known as Y-chromosome microdeletions. So this is where you're looking at the long arm of the Y chromosome, also known as the YQ region. And this is where there are 3 broad regions or subregions known as AZFA, AZFB, and AZFC. So these carry genes that are important for spermatogenesis or sperm production inside your testes. So if they are deleted and they can't be seen by a traditional karyotype, but you require a specific PCR known as STS PCR technique. If these regions are deleted, you end up with a quantitative sperm defect. And this guideline has been there for more than 3 decades at this point. But what we have identified in the past 3 decades is that infertility can also happen because of monogenic disorders. So this is where you might have a biallelic or a monoallelic variant in one of these genes, such as TEX1, DPY19L2, and so on and so forth. But they are not screened even today in routine healthcare. And that's partly because of the high cost that goes along with testing for all of these different genes, as well as karyotype and YQ microdeletion. And because of all of these issues, only 5 to 10% of the patients who actually do come to clinic gets a genetic diagnosis. The majority of the patients remain without a genetic diagnosis. And it's a travesty because providing a genetic diagnosis helps in giving prognosis to these patients. That is to say, whether you should go for a regular IVF, or you might want to go for an ICSI, or you might want to do a testicular sperm extraction procedure. And based on that, you can start giving them personalized medicine in the sense that whether their children will also inherit their infertility down to the next generations. Clearly, having a genetic diagnosis for this disorder is very, very important. Now, my group has been working in the field of infertility since about 2018. And during that period, we published this wonderful paper in Nature identifying a brand new gene known as RBM5, which is where even a single allele of this gene, if it gets knocked out, would lead to a complete spermatogenic arrest. That means no sperms are produced in the testes. Not only that, when we look at the qualitative sperm defects, we actually identified a lot of new candidate genes associated with what is now known as multiple morphological abnormalities of sperm flagellum, MMA for short. It's a bit of a mouthful, but essentially the tail of the sperm is wonky. It's not properly formed, and because of that, the sperms don't travel anywhere. They just keep on going round and round in circle, and that's because of these genes out here. And the reason why we were able to identify all of these genes is because we started shifting from traditional karyotype and STS-PCR based approach to more sequencing based approach. And it's not just our group that started doing that. If you look into the literature, you do identify that exome sequencing is playing a major role in gene discovery and diagnosis, especially when it comes to male infertility. Monogenic forms of infertility are actually on the rise. We do identify a lot of these genes coming through. And in fact, right now, there are 120 single gene disorders for which we have a 100% gene disease relationship. That is to say, if we know that this gene is knocked down, the patient will develop a certain type of infertility phenotype. And I've listed them out here for your reference. Now, one would think, okay, fine, let's just do whole exome sequencing on anything and everything that moves that has infertility. The problem with that approach, or that broad approach, is that your exome panels do have certain deficiencies. If you're not picking the right target enrichment kit, you're not covering the YQ region in the genome, and therefore you'll be missing out on YQ microdeletions. Or if you're not sequencing at a deep enough coverage, then you might miss out on copy number variants, which also occur at a very high frequency in these disorders. Not only that, generally our tendency is that the larger the NGS panels are, our anticipation will be there for higher diagnostic yield. And hopefully by the end of it, I'll prove that that's not really true when it comes to infertility. And of course, we all know if we increase the number of genes that we are screening, the cost per sample also increases quite tremendously. And therefore, if you have a big panel, you require large NGS instruments, you need large overheads, your infrastructure cost and your labor costs suddenly start to shoot up. Now, what we find in India is that Illumina sequencing platform, most notably MySeq on one end, which is the baby instrument, and on the other hand, your NovaSeq X or NovaSeq platforms, they are the most common ones you find in genetic diagnostic lab these days. And therefore, you want a test that should be able to scale anywhere between these two instruments. And that's a key challenge for anybody to design a genetic test these days. And a critical thing is you want to focus on monogenic forms of infertility because that is where you will find majority of your diagnostic yield. And that's exactly what we started doing around 2022. Since 2022-2023, we collated a list of 39 genes that covers 8 common male infertility phenotypes in India. And this was done in tandem with Professor Joris Wiltman, who is now based at University of Edinburgh, Dr. Sheth and Dr. Sheth, who are the heads of Institute of Human Genetics in Ahmedabad, and my former colleague, Shrutika Kale, who actually did a lot of this test development in-house. Our idea was that it will be a targeted gene panel, and it will be designed based on the ACMG guidelines. Yes, ACMG do have guidelines on how you should design a targeted gene panel. Our logic was that it should fit well within Illumina MySeq platform. One shouldn't necessarily be forced to use NovaSeq platform. If they have a baby sequencing platform, the panel should work within that capacity as well. It should be able to detect not just single nucleotide variants, which is fairly easy, but it should also be able to detect copy number variants, that is even a single exon deletion or duplication event, and it should be able to pick up gonosomal aneuploidy, so an event like Klinefelter syndrome, rather accurately. And since we are Indians, and especially I'm Gujarati, I'm fairly thrifty. Therefore, the assay should be low cost and it should be amenable to high throughput. Now, to tick all of those boxes, we went back to a technology way back from 1990s. Back then, this technology was called padlock probes. Today, it's known as single molecule molecular inversion probes, or SMIPs for short. The logic is fairly simple. If this is the region of interest that you want to sequence, you design a probe that would hybridize to either side of your region of interest. You will then simply copy the region of interest using a polymerase, and you would circularize your MIP probe. And now, after a round of amplification, you're ready for sequencing. Now, the beauty of using SMIPs rather than other amplicon-based approaches is that you have something known as a unique molecular barcode. In our case, it was just a 5 nucleotide long degenerate molecular barcode. Essentially, anytime a probe hybridizes against a piece of DNA, it captures that event in the form of that barcode, which allows you to know your PCR duplication rate, your accuracy in variant calling, or how good your DNA quality was to begin with. Essentially, overall accuracy of variant calling increases quite dramatically. Now, these probes are rather expensive to manufacture. They cost about 2 to 2,500 rupee per probe, but the running cost is less than 2 paisa per sample, making them exceptionally cheap. Now, based on this using this technology, so far we have garnered 3 patents, 1 international, 2 national patents, and we have 5 international publications. Our 6th one is actually in the pipeline at the moment. And the way you go about using a technology like this is that when you have a lot of samples coming in, you might want to use a technology like that of Cambrian Bioworks to do an automated preparation or an automated DNA extraction. And once that entire set of DNA is extracted, you're ready for your library prep, which is where you would have a pool of around 1,500 SMIP probes that will target your 39 genes. You would then pool your library, and this entire part takes only a day. It's an overnight step, actually. On the second day, you're ready for pooling your libraries. You now go for sequencing, and we sequence our libraries at 200x depth on Illumina sequencing platforms to ensure that our CNV calling accuracy is absolutely to the point. Once the reads are ready, you would align against a human reference genome. In this case, it was HG19 or GCRH37. And through an automated pipeline, you would detect SNV, CNV, gonosomal aneuploidy, and voila, you're now ready for your reporting, all done within a week's time. And here are the broad examples that I want to give how a technology like this is able to help in improving diagnostics. So here is an example whereby a patient was tall, thin, slender, had azoospermia when we did semen analysis. And when we looked very carefully at the X chromosome, we saw an excessive increase in coverage in that region. When we zoomed in a little more, because there are 2 genes that are present on the X chromosome, we identified that in fact, when we assess the biallelic frequency, we are getting a heterozygous call. Now, a heterozygous call would only come through if you have 2 copies of X chromosome. So then we did even further a deeper analysis to try and see the broad coverage that is there for these 2 genes. And it turns out that compared to all the other samples that were there, there was an excessive coverage, thus showing that this patient actually had a Klinefelter syndrome, that is 47XXY. This is something that we could diagnose fairly easily with our sequencing assay. But we didn't just stop there. We wanted to see if we could be all the way up to the EAA or EMQN standards for when it comes to YQ microdeletion. So these are, as you can see here, these are all the markers that are used for STS-PCR. STS-PCR essentially ensures that your primers bind to these specific loci out here. Therefore, you're amplifying a very specific segment of the DNA. We, in turn, ensure that we are amplifying specific genes that cover these regions. And if there is any deletion or, in fact, a duplication in this region, we will be able to pick up and be in accordance with the EMQN recommended marker. Lo and behold, here is an example whereby this patient actually had deletions of multiple genes. So this is where you have deletions of DAS1, RPS4Y2, BPY2, and so on and so forth. Essentially, you had a deletion of the entire AZFB and AZFC region, which will lead to azoospermia. And in fact, this patient did have azoospermia. We didn't stop even after that. We said, okay, let's take 120 patient samples, whereby we know the clinical phenotype, but we have no idea about the genetic makeup of these individuals, except that the karyotype is normal and they have no microdeletions on the Y chromosome. So are there any monogenic forms of infertility that's affecting them? And in fact, in 12 out of these 120 patients, we did identify biallelic variants in genes that were causing infertility. Therefore, there was an uppage of 10% diagnostically immediately. Indeed, 8 out of these 12 patients had variants in the CFTR gene. Now, when we think about CFTR gene, we generally think about cystic fibrosis. That means you would have a pulmonary function or a pancreatic insufficiency coming through. But in male infertility, or especially with CFTR gene, if one allele has a highly penetrant variant and the second allele has a mild penetrant variant, you don't always have a pulmonary dysfunction. You actually do have what is known as congenital bilateral absence of vas deferens. That means you have a blockage of a particular tube through which your sperms are traveling all the way out. And because of that blockage, it looks like the patient has azoospermia, but in fact, when you do test biopsy, you would find healthy sperms in there. Now, these patients, I must highlight, are very critical because they are missed in the clinic. Nobody does, or very few patients actually undergo ultrasound scan of their testes. So now there is a genetic way to identify these patients, even without having a full clinical workup done on these patients. Beyond that, we did identify 4 patients with biallelic variants in these different genes, and these genes are associated with different types of male infertility. And accordingly, we were able to guide the patient as to what sort of IVF procedure they could go to. So, for example, if it's cystic fibrosis gene that we are dealing with, CFTR gene we are dealing with, then the wife should be screened for CFTR variants to ensure that she's not a carrier of any variants in there, and the infertile male could have test a biopsy, get the sperms, go for IVF, and have a completely healthy child down the line. Now, as I said, I'm a Gujarati. I do care about the costs of doing all of these testing. So the entire cost of doing DNA extraction, library prep, sequencing, and data analysis comes to less than US $70. So about 6,000 odd rupees per sample. I'm going all the way from extracting the DNA all the way down to data analysis. When we talk about sequencing instrument, if you are running MySeq in your lab, you can go anywhere from 2 to 50 samples easily, depending upon which flow cell you want to use and what sort of throughput you want to have. But if you're getting hundreds of samples on a weekly or a monthly basis, then you can very well bump up to NovaSeq, whereby you can absolutely bung together more than 100 samples on a flow cell where you're also doing whole exome or whole genome sequencing. So you can do mix and match sort of work on a NovaSeq platform. And all of this cost is there whilst we are doing sequencing at 200x depth per sample. In terms of data analysis, it takes about 30 to 45 minutes per sample to go all the way from FASTQ file to VCF file. Now, when I'm talking about this particular time period, I'm talking about doing analysis on a laptop that consists of a simple Core i5 13th or 14th generation chip, 16 gig RAM. You don't require a large-scale server to do this sort of work. And this panel, along with the bioinformatics pipeline, is now available for clinical use. So, to sum things up, the old paradigm was whereby you would do karyotyping, it would take about 4 or 5 days. If that's negative, you would now do STS-PCR, which would again take about 3 days. And if that's negative, you might want to do CFTR gene sequencing to rule out if there is any blockage in the tubes. And apart from all of that, all other forms of monogenic infertility were still missing. Now, within a 4-day span from DNA extraction to reporting, you can not only just identify Klinefelter syndrome or other chromosomal aneuploidies, you can also identify AZF microdeletion. You could do CFTR gene sequencing and also cover 17 other monogenic forms of infertility in a single test. So to conclude, male infertility, it's significant healthcare and societal burden. We don't always look at males. We generally tend to look at females, but actually males do carry 50% of the problems in them. Where are we looking? Now, that's a particularly important question because so far we have only been looking at karyotype and Y-chromosome microdeletion, but we need to start moving beyond these two tests. We need to look at monogenic forms of infertility because they have a significant role to play in disease etiology and epidemiology. Using a panel such as the one we have created, you could reconcile SNV, CNV calling, chromosomal aneuploidy calling, all in a single test and get an additional 10% diagnostic yield on top of whatever WHO is recommending as of today. CFTR variants, or biallelic CFTR variants, are actually very, very important when we think about infertility. We don't do detailed clinical workup in these patients, especially when it comes to azoospermic men. Therefore, doing a genetic test gives you an excellent proxy to identify these individuals and ensure that the next generation don't come with a full-blown cystic fibrosis phenotype. In terms of availability, the assay is available for clinical use right now. And with that, all the wonderful people, it's not a one-man show. These wonderful team members are our international collaborator, clinical collaborators, all the organizations, and of course, the funders, GSB team, ICMR, and Gujarat Startup Seed Fund, through which this panel could be developed. And the last thing I want to iterate is that a technology like this is developed fully in India, by India, and most importantly, for India. With that, thank you very much.
Thank you very much, Dr. Harsh. That was a lovely talk. I feel both in terms of the depth of your work as well as the scientific rigor that is involved in the work that you have done so far. It's extremely inspiring to watch. It's sort of difficult to see like such a seamless connection between research as well as patient benefit. And I feel that the kit that you've developed as well as the talk that you've just delivered captured both of these really well. What actually stood out to me personally is how you spoke about reducing costs while simultaneously improving the accuracy of these genetic tests. So that is a very, of course, an important combination that is not just technically challenging to achieve, because the more accurate these tests get, the more expensive they get. But like you rightly said in the very end, is how we're building all this tech in India for India. So to scale it and to make it more accessible, definitely the combination that you spoke about captured it wonderfully. So for everyone else who has joined today, at Cambrian, we've had the pleasure of working with Dr. Harsh and the FRIGE team for over a year and a half now. So he's not just been the user of Manta, but a fantastic supporter throughout this time. He's always very generous with his feedback, his ideas, as well as his time. So we are always genuinely very grateful for your support, Dr. Harsh. To sort of bridge this through, I mean, it's work like this. This is the type of work that sort of reinforces why we do what we do at Cambrian. When we built the Manta, the goal was never to just automate for the sake of automation, right? It was about building these tools that actually enable research to happen faster and more affordably, of course. And even as I mentioned, more consistently across different applications. So whether it is rare disease research or reproductive genomics or molecular oncology, a common thread that we sort of see is that all of these genomics applications have to be scalable for them to deliver any impact across the country. So that is why we built the Manta, so that the researchers can actually go ahead and focus on what is important, designing panels like these, asking the right questions and going ahead and analyzing all of their sequencing data, so that your extraction bit of the workflow is taken care of and you can sort of rely on the machine to do that bit for you, so that you can focus on more analytical things so that your repetitive part of your workflow is automated. So whether it is infection control or molecular oncology or reproductive genomics, the Manta supports all of these workflows in the very first part of the entire process. And how we have designed the Manta is that it works on pre-filled cartridges. So I think some of you who have joined today are already the users of Manta, but for whoever who has joined for the first time, the Manta works on pre-filled cartridges. So the cartridges already have all the extraction buffers that you require for the process. So it has your lysis buffers, your washes, your elution, as well as the magnetic beads. So with the Manta, you can run even a single sample at a time, as in you don't have to fill out the entire batch of 32 samples or 96 samples if that is what you're used to working with. In fact, you can run exactly the number of samples that you get in a day. So with the Manta, like I mentioned, the different types of applications that are accommodated and the different type of samples that are accommodated can be FFP tissue, whole blood. In case of Dr. Harsh's workflow, it is whole blood. And it can be swabs, it can be your stool samples for microbiome testing. So all of these, we sort of enable all of this to happen on the same device by completing the lysis part of the workflow off tech. So whatever sample you're processing, if you're a low or medium throughput lab that is dealing with a lot of different types of samples, you can always extract it using the same device. So you can lyse the samples off deck and load them into their respective cartridges, and will run all of these different sample types on the same run on the machine. Meaning you can run 2 blood samples, 3 saliva samples, 4 FFP samples on the same run on the machine. This is something that we kept in mind while building the device. Now, coming to just a small clip of how the Manta works. I hope all of you all can see my screen. This is the pre-filled cartridge that I was talking about. So you can either run one sample at a time or any number up to 32 samples at a time. Now, if I have to list down just top 4 points of why labs pick Manta, it would be because of the pre-filled cartridges. So you eliminate that time of filling out a deep well plate. You also can run any number of samples, which is a flexible batch size that you have. And on the device, it is a very short runtime of 35 minutes. And of course, is compatible with many different sample types that any lab might be dealing with. Some of the samples that we currently, some of the extraction kits that we currently offer include DNA extraction from whole blood, fresh tissue, FFP tissue, total nucleic acid extraction kit in case of viral samples, saliva DNA isolation kit, DNA isolation from cord blood and stool, as well as RNA isolation from whole blood and FFPE. So these are some of our new launches that I think we spoke about the last time as well in the webinar. All right, now I'll just hand it off to Himani for the closing remarks.
Done. Thank you so much, Dr. Harsh and Diksha, for such an insightful session. And thank you all for attending today. If you would like to continue the conversation or have any follow-up questions, feel free to reach out to us or Dr. Harsh directly. Till the next time, and I hope to see you all on our next webinars as well. Have a great day ahead.